share with us your presentation because we lost it now?
Yes, I just put it back. So thanks again for having me, and greetings from New Zealand. I'm excited to share with you some of our past and current research. And the first part of the talk where I'm going to talk about infant gut microbiomes is actually most of the work was done back in the Broad Institute of MIT and Harvard, where I used to be before Liggins. And then I'm going to wrap up with some current work at Liggins, which is related to bacteriophages in fecal microbial transplantations. But let's get started with the infant microbiomes now. And for this audience, I probably won't have to motivate the importance of the gut microbiomes a lot, but I will just use this slide to say that there are obviously a number of different things affecting this dynamic development succession of infant gut microbiomes. And we usually kind of say in introductory type sentence that the infant gut develops to resemble adult composition during the first 3 years of life. Now it's becoming more and more clear that even the microbiome roughly at age 10 in adolescence, it's not yet similar to the adult microbiome, so we should probably kind of update this view a little bit. I have been too lazy to update my slides so far, but as I said, I should do that as well. Well, now type 1 diabetes is an autoimmune disease, and the rates of autoimmune diseases have been increasing together with urbanization, increasing levels of sanitation, etc. This figure here is from a very fresh review on type 1 diabetes, and you can see that the rate has increased since 1960s in many Western countries. And researchers have given this phenomenon many names. They have first of all postulated that these two are interconnected, so that the changing lifestyle is actually possibly causing the changing disease incidence, and then named it hygiene hypothesis, biodiversity hypothesis, old friend hypothesis. There are many such names. And I think these names are actually trying to refer to at least partly to the same things, that we are basically losing some of our commensal microbes. And there might be some reasons we now know to actually believe this hypothesis is true. So first of all, caesarean sections are becoming more and more common, and we now know that it in a way precludes the microbial transmission at birth. Then we use a lot of antimicrobials throughout life, and this may interfere with the microbial communities on our bodies, and also may have intergenerational effects that kind of build up. Then our modern diets contain lots of highly processed foods, and these foods are obviously also food for the microbes. And finally, we live in cities, highly built environments, and we are in these cities, we are obviously exposed to very different set of environmental microbes compared to, for example, living on a farm. Well, then more specifically, what do we know about associations between microbiomes and type 1 diabetes, which is the autoimmune disease of interest here? So early human cohort studies have found, and these are common findings with multiple studies, that Bacteroidetes species seem to be increased in young babies if they later develop type 1 diabetes, T1D, or islet autoimmunity, which is a pre-diagnostic condition of type 1 diabetes. So basically certain autoantibodies start appearing in the blood, and they predict the development of the disease. People also notice that there might be a deficiency of short-chain fatty acid producing bacteria in disease in babies later developing the disease. And then this was actually further studied in a mouse model. Non-obese diabetic mouse is the most used mouse model for diabetes, and or type 1 diabetes specifically. And recently, Marino et al. Noticed that you can actually protect these mice from autoimmune diabetes using short-chain fatty acid releasing diet. So when we started studying the infant gut microbiomes in immune development roughly 5-6 years ago now. We first of all asked, can we see any aspects of the microbiome that might be important for the immune development in very early infancy and also in childhood? And then more specifically, are there any changes in the intestinal microbiome that predispose children to type 1 diabetes? And we set out to look into these questions in a cohort first in this area in Northern Europe. And here in Finland, the incidence of type 1 diabetes is highest globally for mostly unknown reasons. So that's basically one of the driving motivations of this location. And even more intriguingly, here in bordering Russian Karelia, where the population is genetically very similar to Finns, the rates of type 1 diabetes and also other autoimmune diseases are 6-fold lower. So there is a very large discrepancy just across the border. And then Estonia here, south of Finland, is in a way scientifically intriguing transitional case. So up until the 90s, they used to be under Soviet oppression, and they had similar low rates of diabetes and other autoimmune diseases to Russian Karelia right now. But then when they got their independence back and their lifestyle started shifting very rapidly towards Western lifestyle, the incidence of these diseases also started increasing. And now just in 30 years, they have reached similar levels of type 1 diabetes compared to Finland. And this rapid change has been very well documented by our collaborators in Tartu and elsewhere in Estonia as well. So this basically hints that there is something environmental going on. There are environmental triggers to the disease. And microbiome is obviously just one possibility. Well, we started screening newborn infants in these 3 different cities to first of all control the genetics. So there are very well-known genetic risk factors to type 1 diabetes. But here we just wanted to study the individuals who have increased risk to the disease so that we basically could remove the genetic component of the disease. And only families with babies with HLA conferred susceptibility to autoimmunity were invited to join the study. And finally, we were able to collect this cohort of 74 individuals from each city. And here you can just basically see the distribution of different risk genotypes. And first of all, there are no neutral genotypes. And the distribution of other risk levels is similar between the countries here. So basically, by genetic predisposition only, these babies should have similar rates of autoimmune disease and allergies as well. But then we collected rich set of different biosamples, and from the serum samples we can actually follow these autoantibodies that precede type 1 diabetes development. And now if we looked at when we looked at the serum samples at 2 years of age, and here the top bar shows the percentage of babies who had at least one circulating autoantibody predicting type 1 diabetes. There were more than 10% of such babies in both Finland and Estonia, but only less than 2% in Russian cohort. So basically this is already hygiene hypothesis in action issue, which now these babies with similar genetic risk show differences in their phenotypes. And similar differences in certain circulating IgEs specific to these allergens, so egg and milk allergy or IgEs predicting that allergy were very common in the Finnish and Estonian subcohorts, but more rare in Russians. And now looking at these numbers, you should remember that this is genetically predisposed population, so these diseases are obviously not this common in Finland or Estonia, but in such a subpopulation we can see very high incidence of such phenotypes. But we then also collected monthly stool samples from all these babies during the first 3 years of life to follow their gut microbiomes. And we we have sequenced, or at this point we sequenced roughly 1,600 stool samples using 16S RNA sequencing, and also almost 800 samples using more detailed metagenomic sequencing. And let's start by looking first into the 16S sequencing data. So when we look at the PCOA ordination here, each point is a stool sample, and the closer together they are, the more similar the gut microbiomes. You can see that the age here is perhaps non-surprisingly the driving factor of the ordinations or early samples here on the left, and the later samples on the right. But if we color by country now, we can see that there is perhaps also a shift in by country, such that especially the early Russian samples seem to cluster here in the bottom left corner. And now looking at the phylum level composition in time, so that birth early samples are here on the left and going up till 2 years of age here, and Russia in the bottom first. You can notice that Russia seem to have more Actinobacteria, which also contain the Bifidobacteria genus here in the bottom, and then Russians and Finns instead seem to have more Bacteroidetes phylum containing the Bacteroidetes genus, especially during this early first year of life, which is obviously the time of breastfeeding as well. Well, now moving to just slightly different view of this first, and this is actually also moving to use the metagenomic data. So just allows kind of diving down to the species level, but if we first look at those genera I mentioned using the metagenomic data, and now these bars show fold change between Finns and Russians. So basically there was a pronounced change in Bacteroidetes genus, so that Finns have more Bacteroidetes, especially during first year, and in turn Russians had more Bifidobacterium. And some species in these two genera are here. So Bacteroidetes dorei was actually the most differing Bacteroides species between these two countries, again such that Finns had more Bacteroides dorei. And this was a species that I mentioned in the beginning that some previous studies had associated already higher Bacteroides in type 1 diabetes, and this was actually the species that was found to be associated with type 1 diabetes pathogenesis already previously. And then there were some differences in Bifidobacteria. So especially Bifidobacterium bifidum seemed to be more common in Russia, but one species, Bifidobacterium breve, was still more common in Finns compared to Russians. Well, one of the first things we now wanted to look using the metagenomic data, which obviously also allows us to look at the bacterial functions and the genes that encode these functions. We want to look at the human milk metabolism. And human milk very specifically contains these complex sugars, human milk oligosaccharides, which constitute a major energy source for the bacteria in the gut. So humans actually, we don't have enzymes to break up these complex sugars, but bacteria have to first degrade these sugars in order to get any energy for us as hosts. And Bifidobacterium and Bacteroidetes are known to be two main groups of bacteria that metabolize these complex HMOs. So we basically wanted to measure the genes that conduct HMO utilization and then assign them to the genomes they belong to. And kind of very high level summary plot here, just looking at the average amounts in reads per kilobase per millions of reads of these HMO utilizing genes per country. And basically the height of this bar is the average abundance, but there was no big differences between the countries. But then when we look at the coloring of these bars, so basically on what genomes these genes were assigned to, there is a big difference. So most of the known origin of these genes in Russians were on Bifidobacterium, but in Finland and Estonia they were carried by Bacteroides species. So it seems that the HMO metabolism was basically conducted by different species in these babies. But then we wanted to ask more generally whether any other bacterial functions that would be different between the countries. And just because the taxonomic composition was so different, it was kind of easy to predict that there might be also many functions. And indeed, there were more than 100 different gene ontology terms, for example, that were differing between the countries. Here is just a top 10 differently abundant functional pathways. So on the top 5, for example, these are microbial functions that were more common in the Finnish microbiomes, and in the bottom, something that is more common in Russia. And here we noticed 2 intriguing pathways. Both are related to lipopolysaccharides. So lipopolysaccharide is a surface protein of a surface molecule of all gram-negative bacteria, and lipid A is an immunomodulatory subcompartment of this molecule. And now that if we first look at how our metagenomic data predicted LPS production in these microbial communities, we can see that there was especially big difference again during this early time frame. So basically now just using lipid A biosynthesis genes here as a proxy for LPS abundance, we can say that there is much more LPS in Finnish and Estonian microbiomes in early life. Well, we then wanted to know, obviously, what species again are producing LPS in these microbiomes. And the first usual suspect is E. coli. Its LPS is very well studied. But now when we looked at the abundance, this is just showing the relative abundance of E. coli. There was no big differences between the countries. So it's known that E. coli is more common in very early life. It kind of blooms occasionally in early infant gut, and then it kind of disappears over time. But again, there were no differences in abundance of E. coli. But then when we looked at other, here top 10 species that were predicted to produce LPS, there were more pronounced differences. So there were actually mostly these Bacteroidetes species that we just discussed in terms of the human milk. And now if we look at the abundance of these species first in Russia, they kind of colonize slowly so that they become more abundant only at roughly 2 years of age. But in Finns and Estonians, these species were highly abundant from early on. And at this point, we were very lucky to get 2 researchers who were actually specialized in LPS and immunology to join the study. So Eva de Hensel and Thomas Cullen from Novartis Institute of Biomedical Techniques, and they set out to first culture all these bacteria and then purify LPS from them so that we could do more mechanistic experiments. And now that I talk to you about some of these experiments, I will be mostly focusing on 2 different species. First one, E. coli, is of course the prototypic LPS producer, and its LPS has been very well characterized. And then I will also talk about Bacteroidetes dorei. So that was the species that had already previously been associated with type 1 diabetes pathogenesis, and we also found that it was most differently abundant between Finns and Russia from all Bacteroidetes species. So let's first look at the LPS structure a little bit. So lipid A here in the bottom of the whole LPS molecule is known to be responsible for the immunostimulatory properties. It's basically recognized by the TLR4 receptors in human immune cells. And it's also known that the number of acyl chains, so these little wiggles here in the bottom, it affects the immune activation. And E. coli LPS has 6 acyl chains. It's hexa-acylated, shown here in more closer look. And by mass spectrometry now we were able to verify that the LPS Eva and Thomas purified from their cultures was exactly this known molecule. Then they did the same mass spectrometry experiment for the Bacteroidetes dorei LPS, and this produced two different peaks. So these peaks predict two different lipid A structures, basically tetra and penta-acylated structures. So this, as I said, it's known that the number of acyl chains is associated with the immune activation. So this predicts weaker immune response from these types of LPSs. And now Eva wanted to look into this further. So she first used this reporter cell line, basically NF-kappa B reporter cell line, which reports TLR4 signaling. Lipid A is recognized by TLR4. And now on these heat maps, the luciferase signal first is shown on color intensity, and then the x-axis is the dose of the LPS used. And here in the bottom, shown first the E. coli LPS, there was a robust luciferase response, basically TLR4 signaling with almost all the LPS doses ever used. But then when she repeated this with Bacteroidetes dorei here in the top, there was a very weak luciferase response even in the largest doses. And then with all the other LPSs from these other Bacteroides species, there was a response between these two extremes. Then Eva moved to use human PBMCs, peripheral blood mononuclear cells, and measure different inflammatory cytokines. So now this is basically measuring human immune response to these molecules. And there was a similar trend. So very robust immune response, cytokine response with all the E. coli LPS doses used, but no immune response at all with these Bacteroides dorei and also Bacteroides vulgatus LPSs. So it's fair to say that these two LPS types are basically immunologically silent, at least in these experiments. Well, this is of course very simplistic setting. So we just expose individual human immune cell types to one type of LPS at a time. So we wanted to get these experiments a little bit closer to what actually happens to gut. So first of all these different LPS types are mixing together in the gut to complex cocktails. So to kind of simulate that, we first did some co-stimulation assays, and basically using E. coli LPS as a kind of a control here, and then adding different doses of B. dorei LPS on top of that. Now these are the two extremes from the previous plot. And now looking at TNF-alpha, one of the cytokines, its concentration. So here is the baseline level now with E. coli LPS only. But now when we add different doses of B. dorei LPS together with that, we start to get dampening of the signal. So basically B. dorei LPS antagonizes this immune stimulation by E. coli LPS. It's able to silence the immune signaling. But finally, to get even more close to something people may call immune development or immune maturation, there is a model called endotoxin tolerance. And what this means is that if you expose certain human monocytes to LPS, they seem to have memory in a sense that any consecutive exposures give weaker immune response. So they seem to remember that they were already exposed to these LPSs. So basically, if you first stimulate primary monocytes with LPS, then let them rest for 3 days, and then challenge. This second stimulation is now called challenge. Then you get weaker response in challenge. This is basically shown here. So if you don't use any dose in induction, this is the baseline TNF-alpha response. But as you increase the dose in induction here, then the response in challenge is weaker. So this is endotoxin tolerance as it is described. But now, if we change this LPS used here in induction to B. dorei LPS, basically testing whether B. dorei LPS is able to elicit or induce endotoxin tolerance. We still stimulate in the challenge with E. coli to have comparable results. And we can see that B. dorei LPS is actually only a weak inducer of endotoxin tolerance. So again, there is a difference between B. dorei and E. coli LPS, and this could even be one mechanism whereby the B. dorei LPS is precluding the immune development in humans. Well, at this point, we thought we had nice set of human data and wanted to keep it as such, but one of our reviewers, when we were trying to get these results published, wanted to also get animal models included. So that's when we also did an experiment with the NOD, non-obese diabetic mouse model, and again it's the most commonly used model system for type 1 diabetes, and it is known in this model that you can delay and reduce the overall incidence of type 1 or the diabetes in these mice by IP injections of E. coli LPS. And we were able to reproduce that. So basically, the arrows here show the injections of LPS in these mice, and then the control group is here. So the incidence at week 18 was 50% diabetic in the control mice, but the E. coli LPS injected mice had lower incidence, less than 25%. But then if B. dorei LPS was used, there was no reduction in the diabetes incidence, at least at week 18. So this basically suggests that even more, if you believe in animal models, that this actually precludes somehow the immune development, so that these animals get more diabetic. All right, let's summarize a little bit what we have learned so far. So we studied these two very contrasting populations in Finland and Estonia compared to Russians, and first saw that they have very different microbiomes, gut microbiomes in early life, and especially those bacteria that seem to be responsible for the human milk oligosaccharide metabolism. So many bifidobacteria in Russians, which were in a way replaced by Bacteroidetes species in Finland and Estonia. And then this has certain effects in the LPS exposures, such that Russians are mostly exposed to LPS from E. coli, and whereas Finns and Estonians have this more complex cocktail of different LPSs, and importantly here they contain LPS from Bacteroidetes species, which seem to, for example, fail to induce endotoxin tolerance. So by this route or other routes, this may increase autoimmune activity in these children. So now at this point, you may ask, and we have asked as well, whether there would be a way to start thinking about microbiome modulation and how to shift microbiomes, if you wish. It's always, it takes certain courage to start thinking about such interventions, but here it's actually in a way low bar, because we know that there are some bifidobacterial probiotics, and people ask whether we could actually shift the early microbiomes in Finns and Estonians towards something that Russians have here. Hold this thought a little bit. I will talk about another large infant cohort first, and we kind of try to gather some supporting evidence from those data on the way, and then we will come back to some data with DIABIMMUNE and also with some from future plans. But the other cohort I'm going to talk about is called TEDDY. So TEDI is the largest type 1 diabetes study to date. It's a collaboration between different institutes in Europe and US. It's coordinated from the University of South Florida. So TEDI has clinical centers in Finland, Sweden, and in Germany, in Europe, and in 3 different US states, Washington, Georgia, and Colorado. And TEDI also has collected a vast array of different biosamples. We were responsible for the microbiome analysis only, so that's what I'm going to talk about here. And the study design in the microbiome arm was actually very similar to DIABIMMUNE. So TEDI also collected monthly stool samples a little bit further. So they followed the babies and children with stool sampling until they developed the disease up to at least 5 years of age. TEDI is also larger. So in TEDI, we had at the time of fixing this data set, we already had 100 children with diagnosed type 1 diabetes. Then they had paired controls such that the controls came from the same clinical center and were of the same sex. So we matched also the stool sampling with the controls such that the sequencing of the stool sample stops wherever their matched case had the disease. And similar in the seroconverted, this is the islet autoimmunity cohort now, when they start having the circulating autoantibodies. So we have 267 seroconverted children, and also similarly with paired controls. This is overview of the data. So in TEDI, we had more than 10,000 samples with metagenomic sequencing data, all shown here. X-axis shows the age, and then each row is a subject. So here in the bottom right, for example, you can see all the children with diagnosed type 1 diabetes. And you may notice here that there's a wide range of different age of diagnosis. The earliest babies are diagnosed before they are turned to 1 year of age, and then the latest diagnoses are here at 5 years of age. So this also has marked differences on how long they are followed by their stool sampling, and obviously you can see now the same difference in controls, because we stopped following the controls as well if their matched case gets the disease. And this matching is obviously done after they get the disease, so we don't have any matching before anyone is diagnosed. And similar to the seroconverters here. Okay, well, there is lots of data out on TEDI, and I'm not going to talk about all the different aspects. We have analyzed the microbiomes. Now I'm going to focus on some very specific evidence, but you can read on the microbiome on these 2 citations here. And one of the key things we noticed early on, that the microbiome is highly dynamic, highly subject specific, and something that Chris Stewart did in this other paper is he was actually doing Dirichlet multinomial models to kind of develop this phase model of the microbiome development in early life. But what we can see from this principal coordinate ordination is that there was also some intriguing early heterogeneity. So basically, if we look at these red samples here, which are the earliest samples, there is some intriguing clustering happening. And these clusters were mostly actually due to different bifidobacteria species dominating the gut communities. Also one cluster of dominating proteobacteria, which was more common after antibiotic treatments. But we actually wanted to do a little experiment on this. So we just grew these different bifidobacterial species in vitro with various low nutrient media, and it seemed that these different growth patterns may be caused by different substrate preferences in bifido species. So they basically show very different growth patterns depending on the media. Well, as I mentioned, we learned that the early gut microbiomes are highly heterogeneous, and even if Teddy collected high amounts of different early life metadata, so to speak, different life events, all those information together could explain less than 10% of the variance in the microbiome data, whereas the subject IDs only explain more than 20% of the variation. So basically, this is in a way quantification of how personal the microbiomes are. We noted that cessation of the breastfeeding rather than introduction of the solid food was maybe the biggest shift in the microbiome during the early life, and also noted that there were large geographic differences, basically differences between the clinical centers. Well, now I picked up this result here because we are going to talk about bifidobacteria again, and this was one of the groups that were different in DIABIMMUNE between the countries as well. And different or previous studies have studied this in detail as well, but in TEDI data we also noticed that bifidobacteria seem to be highly susceptible to antibiotics. So whenever you have antibiotic perturbans in TEDI, and we have stool samples 1 month apart, so whenever a pair of stool samples have antibiotics in between, we can ask what are some of the species that were decreased the most after such perturbation. And bifidobacteria come on top of that list. So basically saying that always when you give these babies a prescription of antibiotics, something else tries to overgrow their commensal bifidobacteria. And also, in a way, a side effect of this was that the antibiotic perturbations were larger in early months. And this is because in early months, of course, bifidobacteria are more common. Well, then we, of course, analyzed and compared these cases and controls, wanted to find taxa and functions that were associated with the disease again. Perhaps due to some of those properties I tried to show on previous slides, the subject specificity, heterogeneity, differences between clinical centers, we did not find any super statistically significant differences in cases and controls. We had a list of nominally significant and borderline significant results though, and some of the species that seem to be associated with controls, basically more abundant in controls, are known probiotics, so Lactobacillus rhamnosus, for example, here, Streptococcus thermophilus as well used in milk products, and Lactococcus lactis. And then otherwise, some so-called pathobionts or species were more common, such as Streptococcus. This group of Mitis oralis and Pneumoniae was more common in cases. And when we looked at the functions, similar was true, so there was no super strong, statistically speaking, signal, but there was a common theme here. So many short-chain fatty acid release and meta release related pathways seem to be increased in controls again. So this is now concordant with the previous studies again, and also concordant with the work in these mouse models. But I want to highlight one of the probiotic species now, taxa I mentioned. So there was actually a previous epidemiological investigation in TEDI looking at the self-reported early feeding data in TEDI, and they found that early probiotic supplementation, again self-reported, may reduce the risk of islet autoimmunity. Now, when we looked at, we did not know or collect the information what probiotic strains these families were actually supplementing the babies with, but we saw a difference in Lactobacillus rhamnosus, as I showed you on previous slide. So this was more common in islet autoimmunity controls compared to the cases, and especially early on. So this self-reported usage was also early probiotic usage. And also it was concordant with the prevalence of Lactobacillus rhamnosus. So basically, when they reported early probiotics, they had more commonly, we saw this species in their guts. Now, another commonly used probiotic is Bifidobacterium longum subspecies infantis, or only Bifidobacterium infantis. This is also, just like many probiotics, isolated from human gut. And it's actually this symbiotic species that has co-evolved with us humans to thrive during breastfeeding. So it has this very intricate system for intracellular human milk oligosaccharide metabolism, so that it can avoid cross-feeding these complex molecules with other gut species. But it seems that this is disappearing from our Western populations. And I also have some data on this to show you. It has this very characteristic HMO gene cluster characterized by other groups again. So we are going to use this a little bit to try to follow this species in our data. But first, there was very intriguing trial on this probiotic just a little bit ago, actually 3 years ago. So in this trial, they basically showed that early supplementation of this Bifidobacterium infantis during breastfeeding can actually reconstruct the early microbiome. They, in a way, proved the engraftment of the species, which is relatively uncommon for probiotics to actually engraft, persist after the supplementation is stopped. So here, if you look at these graphs, the supplementation actually stopped here at day 28, but in this case group, the bifidobacterial abundance continued at least up to 60 days of age, whereas the control group looks very, very different. And they later actually released data that this difference was still true at 1 year of age. So it seems to be a very persistent change. So now in our metagenomic data, now I'm switching back to DIABIMMUNE cohort as I promised. We have tried to follow Bifidobacterium infantis as well. And because it's a subspecies of Bifidobacterium longum, it's slightly untrivial to try to detect that in the metagenomic data, but we can, for example, do single nucleotide polymorphism based strain calling, which this phylogenetic tree here is based on. So this is basically all the Bifidobacterium longum strains we saw in DIABIMMUNE data. And here the dots show that the reference genomes among all these other strains that are from the metagenomes. And this reference genome here is the Bifidobacterium infantis. So we kind of hypothesized first that this is the clade depicting Bifidobacterium infantis. And then when we look at the HMO gene cluster I just showed you on the other slide, and independent of this phylogeny, we track those genes in the metagenomes. This actually very well agrees with this SNP-based phylogeny. So in these samples here on the left, these HMO degrading genes, this gene cluster seems to be also present. So this is very clearly the Bifidobacterium infantis subspecies. And we could then ask, for example, does there seem to be any competitive advantage in a way that can these infantis strains grow on higher relative abundances during breastfeeding compared to these other longum strains? And on average, this seems to be true. So here on each country separately, the purple average is basically higher compared to the longum strains, other longum strains. And people commonly ask, actually, is this difference in Bifidobacteria due to differences in breastfeeding? Do the Russians breastfeed more, breastfeed longer? But actually, the contrary is true. So in this cohort, at least, Finns and Estonians had, on average, longer breastfeeding than Russians. So more bifidos in Russians is basically not explained by breastfeeding. And now looking at the Bifidobacterium infantis prevalence, we can ask, does it look like it's disappearing in Finland, and only 10% of Finns in this cohort had Bifidobacterium infantis in at least one of their stool samples. So basically it seems that this species is already very, very rare in Finland, but other cohorts from Asian countries, for example, had shown that this is basically a strain carried by all babies. So there is a sharp contrast here again. Well, now we are ready to wrap this up a little bit. So we started with data showing that Bacteroidetes species have been basically replaced somehow by Bifidobacterium in early life in Finnish and Estonian microbiomes. And this may predispose these babies to type 1 diabetes if they have the genetic predisposition as well. Then we know that there's this commercially available Bifidobacterium infantis probiotic that is persistently engrafting in breastfed infants. And we actually know that giving this probiotic may also cause a major shift in the whole microbial community. Then we know that early probiotics may protect from islet autoimmunity from both of these, or first of all, this epidemiological investigation, and also from the metagenomic data from Teddy's study. So this has led us and others as well to speculate whether it would benefit to at least pilot Bifidobacterium infantis supplementation in genetically predisposed Finnish babies, and this would be done as islet autoimmunity as the primary outcome. So we are currently planning such a pilot first to kind of try this idea. It's obviously very expensive study to conduct. Okay, I don't know if I have 5 more minutes to talk about our work at Liggins, or what's the...
Yeah, sure, we still have 10 minutes.
Okay, great. So then I'm gonna switch gears completely, and now, as I promised, I'm gonna talk about work at Liggins. So one of the exciting studies you may already have heard when Justin was visiting, but GutBugs trial, which is a placebo-controlled trial to test the efficacy of fecal microbiota transplantation in obesity. And this is led by Justin O'Sullivan and Wayne Cutfield here at Liggins. So in GutBugs, our goal was to recruit 100 obese adolescents to receive FMT or placebo capsules. And these material for the FMT capsules came from 9 healthy donors. It was sex matched, such that females only got stool from females and males from males. We had 4 female donors and 5 male donors, and we always mixed the stool in the capsules so that all recipients got stool from all the donors from the same sex. Now the part I'm going to talk about is the microbiome part again. Justin is going to talk about this probably in 1 month when he gives his talk in this series, and he may also talk about the primary outcomes which were related to weight loss and other clinical parameters. But for the microbiome part, we also collected stool samples at baseline before the intervention, and then 6, 12, and 26 weeks after the FMT treatment. And now if we just look at some key measures of the microbiome, this is unpublished. I'm just going to give you some teasers here. But people often look at the alpha diversity, for example. And we noticed a shift in alpha diversities in females, but not in males. So basically here, if you look at in the middle of the female graph first, you have the FMT recipients, and red or orange is their baseline diversity, and green is the diversity at week 6. And there was a significant shift upwards. No shift in placebos in either sexes, and no shift also in males receiving the FMTs. Now if we look at the beta diversities, a little bit looking at the shift after the treatment, and now comparing to the donor profiles, basically 1 by 1. So here 0 means no shift towards the donors whatsoever, and anything positive means that the recipients actually shifted towards the donors. And here now we can break this down by the different donors, by different colors. And in females, this one donor seemed to very clearly stand out. So basically all the recipients, even though they received stool from all these 4 donors, they shifted towards this one donor only. Males didn't have similar phenomenon. So it seems that we may have one so-called super donor in the female donors here. Well, this is my final slide, and I promised to talk about bacteriophages. Now there is some intriguing early data and speculations that, and these stem from the studies showing that actually fecal filtrate with no bacteria also alleviates the Clostridium difficile infection. So basically, the FMD is still efficient even if you take out the bacteria. And this has obviously led people to ask, so what are the effective agents then in stool? And one of these agents could be viruses, maybe phages. So phages is actually majority of the viruses in any stools. And they're also gaining more attention now in more wide fashion as well. So more and more people are studying phages in general. But to me, it's very intriguing, and it will be very intriguing to study the phages in this FMD setting. So I now have a new grant to look into gut viromes in gut bugs trial together with the Forrest rover from San Diego, who is a phage expert. So we basically do phage enrichments and viral enrichments to these stool samples in order to take a very detailed look in the gut viral communities. And there will be also some intriguing basic biology to investigate phage bacteria interactions, etc. And then obviously to look at the phage transfer and the role of the phages in bacterial engraftment and in these community shifts related to FMD. All right, but I'm gonna wrap it up here. I want to thank my mentors, supervisors at the Liggins, Justin O'Sullivan, Wayne Cutfield, and Frank Bloomfeld, and 2 of our microbiome students, Brooke and Clara. Both DIABIMMUNE and TEDdy studies have large international groups involved, so there are too many names to list here, but I thank all those consortiums as well, and I thank you for your attention. Thank you.
Let's thank Tommi. And we have 5 minutes for questions. I don't know if Tommi is willing to stay a little longer if we have more than that, but thank you very much for a very good presentation, Tommi.
Yes, I'm definitely happy to stay if people have questions.
Questions?
So I was wondering with the different populations, looking at the Estonians, the Finns, and the Russians there, when you have that shift towards Bacteroidetes from different bacteria, is that representing kind of maybe a premature development of the microbiome towards more of the adult state, or is it just sort of an alternate infant state that still changes just as much as it goes towards adulthood, but in a different way?
Yeah, thanks. Very, very insightful and interesting question. It could be, I think it could be something. Premature is kind of a strong word, but yes, it definitely looks that the microbiomes in Finns and Estonians, they are more similar to adult microbiomes in a way. Bacteroidetes are very common in adult gut as well, and they also have higher diversity compared to the Russians early on. Bifidobacteria, if they are present, especially if Bifidobacterium infantis is present, they tend to dominate the community, so they basically associate with very low diversity as well. But whenever you get colonized with these Bacteroidetes, it also it's implicated in higher diversity. So it's kind of it's hard to say what's the chicken and egg, so to speak, and whether it means that it's the adult microbiome, whether there are certain species missing, certain keystone species missing, affecting the community dynamics. But you could certainly put it in that way. I think that's fair.
I have one question. So I believe you did match Finnish and Russian moms and babies for like lifestyle and things like breastfeeding and C-section. Did you see differences there? Was that an exclusion criteria on your study?
Good question. So our only strict exclusion criteria was the genotyping. So basically we wanted them to be genetically susceptible, but we did collect all this information about C-sections, breastfeeding, other early feeding. There was actually very detailed questionnaires on diet and early feeding. There are no striking differences between the countries. For example, there was only a handful of C-sections in all countries. I think 5 in Russia and 4 in Finland or something on that scale. The breastfeeding, as I showed, was on average a little bit longer in Finland and Estonia compared to Russia, but no striking differences there either. So Finns on average breastfeed something like 250 days, Russians a little bit less than 200 days. There are certainly differences in the other early food they are introduced to because they come from different countries, different cultures. And actually we were able to find out in one of the later studies, or later, this is described in the latest DIABIMMUNE paper, that the Russians were fed with certain milk product which had the probiotic Bifidobacterium bifidum strain. Yes, Bifidobacterium bifidum. So that probiotic strain basically shows up very clearly in the metagenomic data. More than half of the Russians have identical Bifidobacterium bifidum strain. So that's something that's certainly specific to Russians, for example.
Tommi, thank you very much for your presentation. Feel free to reach out to us. We have many people working in different areas of microbiome. So we look forward for future collaborations with Auckland.
Yeah, thanks very much for having me. And I would be very happy to discuss with any of you and learn more about your research as well. So I think I will be following some of these presentations from the YouTube channel actually, and I hope some of your researchers get to present as well.
Thank you so much.
Thank you.
Thanks. Bye-bye.
Thank you guys for today. If you haven't signed up, please do.
Yeah.